Okay, we want to do a brief overview of cellular respiration. We're not going to go over everything that deals with cellular respiration, but I do want to go back and highlight some things to you just to make sure that you remember some of the things we talked about in class. Okay, if you look below, here's going to be the summary equation for cellular respiration. Glucose, which is C6H12O6, plus oxygen, 6 O2s, will yield the product 6 carbon dioxides plus 6 water molecules and ATP. And remember, with the ATP, there are 36 to 38 molecules of ATP. This next slide is showing you the structure of the mitochondria. And here you can see that the mitochondria has this outer membrane. The outer membrane is smooth. And then if we go to the inner membrane, the inner membrane is also called the cristae. So you'll see this as well. And that's that convoluted membrane. Note that it's not as smooth as the other membrane. It's a convoluted membrane. We know that this is also the location of the electron transport system. You'll notice that between the outer membrane and between the convoluted membrane called the cristae, you have a space, and that space is referred to as the intermembrane space. And this is where, if you remember when we talked about chemiosmosis, this is where we're going to pump the hydrogen ions from the matrix across the cristae into the intermembrane space in order to create that electrochemical gradient across the membrane. Let's continue on. So the reaction groups, if you remember from class, start beginning with glycolysis. And remember, glycolysis takes place in the cytoplasm of the cell. So get a pen here. So remember, we said that it takes place in the cytoplasm. So this is outside of the mitochondria, in the cytoplasm of the cell. The next one is the transition reaction. Remember, the transition reaction links glycolysis over to the next reaction, which is actually the citric acid cycle. And remember, the transition reaction will move us into the matrix of the mitochondria. Let me see, can I grab a pen again? So it will take us into the matrix of the mitochondria. And then you'll see Krebs cycle, also called the citric acid cycle. This will also take place in the matrix of the mitochondria. And then our final reaction, which is where we're going to build all of the ATP, is going to be the electron transport system. And remember, the electron transport system we mentioned just on the previous page, and we said that the electron transport system will take place on the cristae of the mitochondria. Okay? Now, this is what I call cellular respiration in a nutshell. So what I'm going to do is kind of walk you back through the whole oxidation process of glucose, but given the highlights, again, like what we talked about in class. So our beginning substrate, as you can see at the very top of the page, it is glucose. So we said it's glucose, so this is our starting substrate. And we said when we oxidize glucose down to the intermediate, remember the intermediate that we talked about, which was pGal, it says that we would have had to use some ATP in order to get that. So pGal. So when we oxidize glucose down to pGal, it says we had to actually phosphorylate our substrates, which would create our new products. Remember when we put in our first ATP, we created glucose 6-phosphate. Then we put in our second ATP, and we ended up, after an isomerization process, of fructose 1,6-biphosphate. And from there down to pGal, it says we took in, or we used, a -2 ATP. And then it says when we oxidize pGal all the way down to pyruvate, remember we actually used substrate level phosphorylation and took the phosphate groups off of our substrates and put them back onto an enzyme, passed them back to ADP, and it says we created a total of 4 ATP. So if you remember, if we take that -2 and the +4, that's going to give us this total or net for glycolysis of those 2 ATP. Okay, and then so now look at where is this taking place. We said it's taking place where? In the cytoplasm, so outside of the mitochondria. We said, how are we going to get into the mitochondria? We said that the transition reaction would link us to the citric acid cycle and move us into the matrix of the mitochondria. So the starting molecule for the next reaction is going to be the pyruvate. So we said, here's our starting substrate again. It's pyruvate. Now remember we said that pyruvate was a 3-carbon molecule, and what we're going to do is oxidize that down to a 2-carbon molecule. So we're going to take away some carbon, we're going to take away 2 oxygens, and so this is where we're going to liberate that first 2 carbon dioxides, because we've got 2 molecules of pyruvate. Each one is going to lose a carbon dioxide molecule. As you can see, remember the pyruvate will be oxidized and added to this entity that we called coenzyme A, and that's where we're getting this acetyl-CoA from. Okay, and then it also shows you that we're doing what? We're giving away some waste in the form of carbon dioxide. So this is all taking place with our transition reaction. So we said the first reaction up here, this was glycolysis. We said from here to here, we said this is a transition reaction. Transition or preparation reaction. Transition reaction. Okay, and then it says, so the end product for the preparation reaction is the acetyl-CoA, and we said that the acetyl-CoAs are going to move into the citric acid cycle. And we said during the citric acid cycle, this is where we're going to oxidize the 2 carbon molecules finally into the carbon dioxide. So you can see the 2 acetyl-CoAs go in, but what's coming out? The 4 carbon dioxide molecules. It says we also were able to create some energy here. So we didn't create any energy in the transition reaction, but we did create another 2 ATPs during the citric acid cycle. So here's the citric acid cycle, also known as, remember, Krebs cycle. So if you add the 2 totals, it says we make a total of 4 ATP from glycolysis down to the citric acid cycle, Krebs cycle. It says all along the way, it says that our nucleotide coenzymes have been helping us transport energy about the reaction. So they were actually picking up the hydrogen ions and electrons. And for glycolysis, it said we had 2 NADHs that would move up to the electron transport system to drop off hydrogen ions and electrons. During the transition reaction, it says we created another two NADHs, which would also transport their hydrogen ions and electrons down to the electron transport system. If you look at the citric acid cycle, now the citric acid cycle created 6 NADH molecules, plus it created 2 FADH2 molecules, and it's taking those hydrogen ions and electrons down to the electron transport system. If you look, once we move into the electron transport system, remember the formula that we gave in class that said for every NADH that comes in and dumps off its hydrogen ions and electrons, it says he'll make approximately 3 ATPs. And then for every FADH2 molecule that goes in, we're only going to get 2 ATP. Okay? So, as you can see, this is where these values are coming from out here. So, from each of the reactions, we're just multiplying, say, for instance, 6 times 3 ATP molecules is going to give us a total of 18. 2 times 2 is going to give us a total of these 4. And it says for the electron transport system alone, so the ETS alone produces 32 to 34 ATP molecules, and that just depends on what direction we actually take up in this one, whether we actually get 4 or whether we get 6. But it says those two together gives us a total of 36 to 38 ATPs for the whole reaction. I hope this little brief overview gives you an idea of where, and don't forget the electron transport system is taking place where? On the cristae. So we said this is the cristae of the mitochondria, so that convoluted membrane. That's the only one that we didn't mention as to where it was taking place. Hope you guys had a great break.